All right, so we're talking about vision, and the receptors that we utilize or that create vision are called photoreceptors. So the eye itself basically has two parts. There's an anterior and a posterior chamber. They're both fluid filled. And the eye itself is encompassed by three layers. So the first layer is the white of the eye, and that's called the sclera. It's fibrous connective tissue, protective. It's actually an extension of the dura mater, so it's actually part of your brain. The front of this here, which is clear, that's the cornea, where light can enter. And then you have the iris of the eye, the colored part of the eye, the pupil, so the iris, the pupil, and then the lens behind it. The next layer under that sclera is called the choroid. And it is the vascular layer. So, of course, it's full of blood vessels. The layer under that is the retina, and that is the neural layer. All right, there is an area on the eyeball where your visual acuity is. When you're looking at something, this is where the picture is the clearest. So that is called the fovea centralis. So if we look here, this is the fovea blown up into this tiny picture. The fovea, well, the whole retina contains the photoreceptors, and the photoreceptors come in two basic flavors: cones and rods. And in the fovea centralis, the cones are sort of condensed there. So these are cones. Cones are, when you think of cones, think of color vision, color specific vision. Okay, so they're concentrated here in the fovea capitis. So the purple things are cones, the yellow ones are rods. So the light comes in, and the light has to get to the cones and rods to create an image. Okay, so how basically does that happen? So your cones and rods, and we're going to look at one here. So if we have, I can make this right here. So we'll call this a rod, okay? And light comes in on yellow. So light comes in to the cell, the rod cell, okay? And when it comes into a rod cell, it comes into contact with something called rhodopsin. Rhodopsin. Okay, so rhodopsin is a chemical that once light hits it, it breaks it up. So it breaks this rhodopsin into basically two components: opsin and retinol. Once that occurs, that creates a second messenger system, or message, second messenger message. And the second messenger, what happens, it comes down here and it actually closes sodium gates. Doesn't open them, it closes them. This process of the breaking down of rhodopsin is called bleaching. And when that happens, it closes these sodium gates and it creates sort of a hyperpolarized condition on the membrane. So it becomes hyperpolarized here. So normally those gates are open, and when that occurs, the cell that it's connected to, which is a bipolar cell, is supplied with neurotransmitter. So when the gates get closed, it cuts off the neurotransmitter. So gates close, and cuts off neurotransmitter to the bipolar cell. Okay, so when that happens, it makes the bipolar cell fire because it's like, uh-oh, something's wrong. So it fires the bipolar cell. Normally, again, when these channels are open and leaky, it puts a bunch of neurotransmitter there and nothing happens. So once the bleaching occurs and the opsin and retinol come, or the rhodopsin separates into those two, that closes the sodium channels, and the bipolar cell gets cut off its neurotransmitter and it will fire. So where does it fire to and what happens? Well, if we look over here, this is a close-up of the retina cells. Okay, so light comes in here, and it has to get to the photoreceptor cells, the rods and cones. Okay, so once it gets into the rods and cones, the light comes in here, it goes to the rods and cones, and there are certain cells here that kind of help this occur. There are cells called horizontal cells and amacrine cells that help the light to not stimulate, like overstimulate photoreceptors that aren't supposed to be stimulated. It helps to funnel the light into the correct photoreceptor cells. And then from there, it helps the action potential, or once the light gets there, it causes the bipolar cells then, which are here, so bipolar cells in there, to fire. So we have the light, we have bleaching, the bipolar cells fire, and once they fire, that action potential goes into the ganglion cells. So this layer here are the ganglion cells, and if you look at the ganglion cells, which they're similar to, they're basically bipolar cells themselves, but the action potential comes down here, and those the axons converge together, and those axons become the optic nerve. So the action potential goes down the axons of the ganglion cells, which make up the optic nerve, and that goes into the brain over to the occipital lobe. Okay, so this is a very, very gray description of vision. There's way more to it than this, but overall, this is basically what occurs, and this occurs generally at the fovea centralis. Now remember, when you're looking at something straight on, you see, if this is the fovea, you see this clearly. But you can also see things over here too. So those bipolar cells are also going as well. So you can also see vision in the periphery as well as this one, but it won't be as clear as what's happening at the fovea centralis. All right, that about does it for our senses, or what we need to know as far as the special senses. Just keep in mind, these are the senses. The four are, the smell, taste, vision, hearing are the special senses because they have a special receptor, a special cell that takes on the vision. So, again, we have the retina here. Retina, which I can't spell. So the retina in the eye, the cochlea in the ear, and the cells that are, with the olfactory epithelium and the taste buds in the tongue. So that's basically what makes them special compared to our touch receptors, which are general, general senses.